right, let's go ahead and get started. Hello and good morning, everyone, and good afternoon or evening to the folks who may be tuning in from the East Coast or elsewhere in the world. Thank you so much for being here for this Benchline webinar on modern data informatics for cell line development. My name is Stella Howard, and I am a marketing manager here at Benchline, coming to you today from Oakland, California, and I will be moderating today's webinar. So this event will, of course, briefly cover some of Benchline's capabilities for cell line development, but also we'll be diving into some of the bigger data and collaboration challenges we see specific teams and organizations encounter when using this fantastic application. Plus, we'll be getting some advice from some of our experts here that we have today on how to overcome some of these major hurdles. So with that, I would love to go ahead and introduce some of our speakers that we have. Prem Mohanty is one of our senior product marketing managers here at Benchline with over 13 years of industry experience across biopharma R&D and software product marketing. His work here at Benchline now focuses on our solutions programs where he curates internal training and enablement programs for the very wide range of verticals that Benchline can be used in. Thank you so much for joining us today, Prem. How are you and where are you joining us from?
Good morning, Stella. Thank you for that kind introduction. I'm joining from Dublin, California.
Great. And next, I'd like to introduce Wendy Ochoa, who is a scientific solutions consultant here at Benchline, who will be giving our live demo today. With a PhD in biochemistry from the Scripps Research Institute, TSRI, her postdoctoral research focused on structural virology. Wendy is now an expert at curating scientific environments on Benchline for potential clients that specialize in many different verticals. Thank you so much for being here, Wendy. I believe you're joining us from San Diego today.
You are right, Stella. Thank you for the introduction. I am in San Diego.
Awesome. Thanks for being here. And next, I would like to introduce Sean McCormick, who is our team lead on the Benchline Professional Services team and has been leading implementations and developing best practices for Benchline for over 2.5 years now. Prior to his work here, Sean spent 3 years working as a researcher in the therapeutic applications of optogenetics after receiving a master's in biophysics. Thank you, Sean, for being here. How are you and where are you joining us from?
Thanks, Stella. I'm great today and I'm joining from San Francisco. It's good to be here.
Okay, and with that, Prem, do you mind taking us to the agenda? Perfect. Thank you so much. So first, a quick housekeeping item is that we encourage audience engagement. So please feel free to use the Q&A button at any time at the bottom of your screen. That's how we'll be moderating Q&A. Do not use the chat button. And when we arrive at our Q&A break at the end of the webinar, we will be taking some of the questions live, but we'll also be answering some questions in the thread. So for the first 10 minutes of today's webinar, will be Prem covering cell line development, its current state, and some of the major challenges seen, followed by a 15-minute live demo from Wendy that touches on specific cell line development teams. And then next we'll be getting advice from Sean on how to build a successful platform for the application. And then we'll be going into Q&A. So with that, I would love to hand it off to Prem.
Thank you, Stella. Good morning, everyone. As you all know, cell line development is a process of generating large populations of identical cells that are well characterized. Cell lines are used widely across life sciences R&D. Some of the most common applications include biologics production, drug screening and discovery, in vitro assays and testing, especially to study efficacy, safety, and tolerability, and finally for fundamental research use, such as study of cellular pathways and targets. More recently, there has been increasing interest in cell line development. This is primarily driven by the steady stream of novel biologics and biosimilars that are entering the market and require cell lines to produce these molecules. Cell lines also continue to be the go-to platform when it comes to in vitro testing across research. And finally, novel genetic and cell engineering technologies are enabling the bioproduction of complex molecules, which has further expanded the utility of cell lines. Cell line development as we know it today has evolved tremendously over the past. Scientists continue exploring innovative approaches to modulate cells, improve cell line properties, and fundamentally understand the cell biology. Let's take a closer look at each of these factors and some of the associated challenges. Modulating cell lines is complex science. Scientists are exploring a multitude of methods to optimize codons, engineer expression vectors, develop new host systems, and engineer host systems. The challenge here is that the software tools that support the science are often siloed or not up to date. As a result, you might use one tool to run your favorite codon optimization algorithm, while another tool for designing plasmid sequence with selection marker attenuation. And then you have to bring the sequences and the data from these disparate tools into your ALN or registry database to actually do the experiment. Cell line screening strategies and processes continue to be optimized and automated to improve process flexibility, cell line stability and titer, and increase robustness in producing high quality cell lines. Early on in cell line development, you might evaluate different cell line screening strategies such as FACSys-based screening or clone picks, mammalian colony picking, or even evaluate automation or microfluidics for simultaneous high throughput screening. Further downstream, during scale up, you might evaluate multiple approaches such as scale up and scale down modeling and design of experiment to evaluate multiple process parameters. The challenge here is keeping up with the rapid iterations and changing processes as you go through these different stages of cell line development. How do we rapidly iterate while capturing data in a structured manner to still make quick process optimization decisions? And finally, comprehensive characterization continues to be an area of focus to ensure phenotypic and genotypic stability of cells throughout in vitro aging. An increasingly diverse set of orthogonal characterization assays are being used. There are newer methods of characterization, increasing use of lab automation, and continuous testing or monitoring. The key challenge here is to centralize the data from these disparate sources and then present it in such a way that you can quickly search through the data and make timely project or program level decisions. Every cell line development team deserves a solution that will accelerate and support your workflow. There are four core attributes to consider for any digital solution. First, it has to model the science you focus on, which means having the ability to build data models that fit your science. Second, it has to support the processes and workflows that your team performs and have the ability to scale, iterate, and evolve over time. Third, it has to empower users to make both scientific and operational decisions quickly with complete and accurate data. And finally, it has to ensure that users will immediately adopt the solution and recognize the value promised by the solution. The life sciences industry has evolved tremendously over the past decade. We are proud to work with several leading companies that have helped develop new advances. This has helped build up our domain expertise in life sciences and cell line development more specifically. With that, I would like to hand over to my colleague Wendy to show us how Benchline can specifically help with cell line development.
Excellent. Thank you. Thank you, Prem. And hi, everybody. Good morning. As Stella said, my name is Wendy Ochoa, and I am a scientific solutions consultant at Benchline. So we are moving right now to the second part of the webinar, where I'm going to show you through a live demonstration different ways in which Benchline supports the organization, an organization that is working in cell line development. All right, so we see teams struggle with several key challenges. For example, a cell line screening scientist is typically using different software tools for managing incoming requests, performing in silico molecular biology, and screening. And this requires manual effort to transfer and link data across these systems. Similarly, a cell line analytical and testing scientist who runs a bunch of different assays with different instruments from different studies, this can be actually challenging as well to put everything together and analyze all that data. And for the cell banking scientists, the software tools used to track inventory might be disjointed from the ELN that contains experimental context. So this makes lineage tracking quite time consuming. I'm going to divide the presentation into three different sections based on these three teams. The cell screening team, the analytical team, and the cell banking team. I'm going to start with the screening team. This is the team that performs the first phases of the line development. These scientists complete the transfection process and perform initial clone selection, ensuring monoclonality. And they do some analysis to explore the performance of the new cell lines. For a successful transfection, it's important for this team to have access to the full experiment and the sample context related to the experiment they're about to work on. And even if this team doesn't do the in silico design, it is key to have access to the sequence level information and the tools to analyze that information. And the team needs to maintain centralized record of the screening process and the relationship between the parent cell and the polyclonal pools and the monoclonal hits. So I'm going to show you how Benchline can support this team to ensure this transfection and screening process. Okay, right now I have logged into Benchline in my account. Benchline is a cloud-based application, and I'm accessing through my login to an environment that we have configured for this particular presentation. When I log in, I can see some of my current activity. Also a menu here that is going to take me to the different applications in Benchline, but also icons to the main activities like the global create, the global search, where I'm going to find everything that is in my account. And also here an icon for projects that is going to take me to the contents of my ELN. So today, as I'm a cell screening scientist, and my work today is going to be fulfill a request that I received from the molecular biology team to actually prepare a new cell line. So here I can see in this request panel all the different requests, the status of the request, who sent the request, who this request was assigned to. If I click in one of the requests, what I can see all the details of that particular request, all the information the molecular biology team has passed. And I can see information about the experimental steps, but also information about the sample. I have here the cell line that I'm going to transfect, and I have here information about the plasmid that I'm going to use, including information of where to find that particular plasmid. I can, through these links, get all the way to the information, the sequence level information of that plasmid. And I have here now the sequence, 3 different views, and also have access to the molecular biology tools that are going to allow me to maybe explore, modify, and analyze that sequence. But again, I might not be the one doing that work. So what I can do, I can access work that maybe has been done before by the molecular biology team. For example, the analysis or the alignment that was done with the AB1 file that we received from the sequencing process. With the plasmid, I can see also metadata that's allowing me to classify and organize all my information in Benchline. I'm gonna go back to my request, the panel for that request. And so we have agreed as an organization that all experimental records are going to be captured, so we can always go back to those records. And we're using the electronic notebook to actually capture that experimental data. And I'm going to use the ELN to capture the information about the transfection experiment. I'm going to show you one that has been completed already. And here you can see that I have the inputs that come from the request. So I always keep track of that information. And I'm going through the process of the transfection, and I'm adding information of how I prepare the cells, for example, and an image of those cells, how I prepare the reagents, and I have tables that are going to allow me to do the calculations for those reagents, and also the details of that transformation, the conditions, the markers, the selection process. Once I have done the transfection, I have a new cell line, and it's important for the team that we keep track of these new cell lines that we produce. In this sense, I'm going to use a registration table to capture that information of new cell line and add it to our organization database. I have added here information about the parent cell bank and the input plasmid log that I use. and I have created a record of that new cell line. And once I access the information, I can see that the system is helping me to keep track of that lineage and all the relationship with other information that was already in the system. For example, I can see directly the gene that was introduced in this new cell line. I have also a record of the cell banks that have been produced from this new cell line. This has been all entered through the electronic ELN, the ELN. Next step would be isolate the monoclonal cell lines, and I'm gonna do this also through the ELN. I'm gonna keep track of all this information through ELN. And in this case, I'm actually selecting these cell lines visually, so I can use the entries in my notebook to add information manually, or I can transfer information directly from the instrument that I'm using for this selection. Once I have those lines, I register the records in our database through a registration table with all the information that is important for us to keep track of. All the lineage, the parent cell, the monoclonal, so the polyclonal bank, etc. And I'm actually capturing some results, some records that will be used later on. For example, like these images for the process, and even the monoclonality reports that will be used by other teams later. I want to do some initial analysis, and this is what I'm capturing as well, results of this, the viability and the protein concentration. And I can see here that I have analyzed that data right in the platform. So in real time, I can see which of these clones should move forward to the next step. I have expanded those clones, and I have transferred those clones into my physical inventory. So I have now a record of that in my physical inventory. Great. So we're going to move to the next team. And the next team is the cell line analytical and testing team. So this team is responsible for the characterization of the cell lines. And they are going to do experiments like cell line authentication, sterility, cell bank genetic stability. The detailed analysis of this data will allow the team to make a decision on what candidates move downstream. But to analyze this data, this team needs to centralize and standardize the results coming from different places. Ideally, they can evaluate the data right here in the platform to make real-time decisions. And it's not only about understanding the results, but also understanding the team's productivity and the program and testing performance. And this way we'll be able to better allocate resources. I'm going to show you how Benchline can support this team in this process of analysis. Okay, I'm going to go to the project in which we are capturing information for this particular team. As you can see here, most of the experiments that this team does are related to characterization, so authentication, stability testing, productivity. And so we can not only keep track of the experimental data, but we keep track of the results in a way that is going to help us analyzing those results and comparing those results with different testing, with different studies, with different instruments. Here I have an example of an experiment and images from the capillary electrophoresis. And I'm about now to actually capture the values that come from the instrument. And to minimize manual entry and to ensure data integrity, what I'm going to do is I'm going to import the information directly from that file that it was produced by the instrument. And the system is going to capture and compare information in this file with information that I have set up in my platform. And here I have all the data automatically added from that file. We can configure the system, the platform, to fully integrate with liquid handlers and analytical instruments. This way, we will have a very seamless way to, from our notebook, create the input files that we are going to feed these instruments, and a very automatic way to ingest output files that are going to bring information for what happened in the liquid handler or the results from the analytical instruments, and coordinate that information with our notebook, our registry, request, inventory. And yeah, we are capturing this information, but the benefit of capturing this information in a structured way is the fact that now we can query that information. And we can see information that comes from different studies, different projects. And when I get into like maybe analyzing that information, I can see maybe different patterns for cell growth. Oh, I can see all my sterility tests here, productivity, and I can answer questions in real time. For example, I can see here that this cell line might be the best producer of these clones, but it seems that we have a problem with the sterility on those banks. So again, I'm answering questions that we have about this data right here from the platform without the need of going into other software and exporting data. Great, so I'm going to move now to the third team. Last but not least, we're going to talk about the cell bank team. This team supports the entire organization, maintaining cell lines, managing the cell banks, expanding those banks when requested by other teams for experimentation. But for this team, it's essential to track the lineage of the cell lines and have easy access to the history of the master and working banks. This cell is responsible, as we said, of expanding cell banks and setting up passaging experiment. And ideally, these activities are standardized and the team has templates they can use for the process. And the team needs to keep track of the physical inventory. So what a better way to do this than visual dashboards that give this team a real-time picture of the entire collection of and the status of the banks. So let's see how Benchline support this part of the work. I have another folder to collect information from this team. And again, this experiment this team are doing are related to banking mostly, and cell maintenance, and maybe expansion of cell lines. And again, as I mentioned before, ideally we'll have as a team, we have developed some templates to be able to keep doing all this process in a very standard way. So a cell banking scientist that is about to do one of these experiments is going to take advantage of these templates that we have designed. So we ensure that the process is done based on the standard SOPs. And these are different types of templates that we have created. And I'm going to show you one that has been completed based on one of those templates. In this case, we are preparing master banks, and you can see here that I have the steps that I should follow. I have here a table to register the new cell bank, instructions on the freezing process, and I'm adding a table here that is going to help me keeping track of where these banks are going to go in my physical inventory. I have captured the freezing history for the banks, and also some characterization like maybe viability, and I'm capturing as well the passage. And again, by adding all this information this way in the system, what I can see is that my team now can create these dashboards that are going to give us the real-time view of the inventory. And we can see as well, loading. Now we can see also bank activity, what the status of this bank, which ones are available, which ones are reserved. And we can also see, okay, which of the protocols seems to be working better. And also we can follow up and follow the health of those cells with time through a visual dashboard. So we have seen during the presentation how Benchline can support all these three teams with their individual challenges. from screen processing to cell banking generation. But with Benchline, we can optimize not only individual team processes, but we can solve challenges that are related to collaboration and communication between teams.
My name is Sean McCormack, and I'm a team lead on the Benchling Professional Services team. And so I think we all enjoyed Wendy's demo, and what my team is tasked with is taking a new client and onboarding them to Benchling such that they can really benefit from the platform as she's shown. So when we encounter new cell line development teams that are looking to use Benchling, we often find teams that are using many disparate solutions to manage their data. This could mean using in silico tools to create plasmids, create primers, and design primers, as well as other sequences. Perhaps those teams are using spreadsheets to track cell lines, to track transfection vectors, among other things. Of course, we need to record our day-to-day experiments, so these teams will be using paper notebooks or electronic lab notebooks where they can record the day-to-day, and perhaps assay data is being stored in another assay data drive or on spreadsheets as well against the item which is being assayed. For requesting new things be produced or doing data handoffs or just communicating between teams, we often see folks using email, Slack or other communication platforms. And then, of course, there is going to be physical inventory. So folks are going to have freezers or fridges. Often freezers and fridges are backed with a spreadsheet that kind of tells you what's in that freezer or fridge and where it lives. Often they are not, and people are just putting samples on the shelves and hoping that those samples are there when they return. And so what we want to do with Benchling is take these many disparate solutions that we see cell line development teams using and unify them into a single platform. And so a transformation in Benchling will look like your team doing your molecular biology work for the creation of plasmids and sequences in the Benchling platform itself. You can register those plasmid sequences primers directly in the Benchling registry, and you can use the Benchling registry, as Wendy's shown, to track your cell lines, your viruses, your plasmids, and any other data that has significant metadata that you want to know about. You can use templates to record your experiments as opposed to your ELN, but you can also just have standard notebook entries where you can start an experiment from scratch. If you have processes that you're doing over and over again, we do encourage you to use templates because you can set up the experiment, kind of take out the grunt work of setting that experiment up in your notebook each day and just record the pertinent data. We can also use our templates to capture assay data. So you might have an entity that you register, like a cell line. Someone could put in a request for that cell line to be assayed, and another person on the team can open up a template and capture the result data in a notebook entry itself in order to have a complete picture of that cell line. I mentioned request. Benchling requests can replace email or Slack for your communication. As Wendy showed, you can use requests to place a request for a new cell line, place a request for an assay, place a request for a plasmid to be produced. And then for inventory, for each item that you are storing, you're able to say, this cell line lives in this freezer, on this shelf, in this rack, in this specific box. You don't have to get that granular, but you can. What we want to do is kind of save you time from having to dig through the freezer looking for your item. In order to get you on a unified platform, the professional services team is going to do a few things. We're going to start by engaging your organization with initial discovery. And so we'll start by kind of understanding what your organization is. What is the structure? Who's on your team? Who are the scientists? How do you do your work? After that, we'll start talking about configuration requirements. Of course, we know you're a cell line development team, and so we have a basic understanding of what you're trying to do. We really want to know what makes your organization unique and specific so that we can really cater to your use case. This will include understanding your existing research flows, and then, of course, understanding the current state of the data. If we go back to the scientist on the first slide with all of those disparate solutions, we want to know what those are and how you're tracking them so that we can start making a model that's going to capture it all in Benchling efficiently so we can get you on the platform quicker. Once we've understood all this, we'll establish some project management plans and timelines to get us started. Into the configuration and training stage, we'll start by training your team on the Benchling platform so that you're familiar enough with it to help make decisions as we plan. Then we'll design some ideal workflows to solve your pain points and capture your data, and then configure a Benchling registry results and the inventory using our cell line development best practices. So our teams worked very hard to develop best practices for many R&D functions, and cell line development is one of them. To this end, we have data models that work really well for capturing the entities and schemas that will be needed for cell line development. And we can use that as a starting point. So we spend less time on like the initial 50% or so of configuration that's consistent across many cell line development groups. And we can spend a lot more time on your client-specific specifications to make sure that the Benchling solution really fits you. Following this, we'll do some iterative testing and feedback. So we'll build templates that are modeling each of your processes. Your team throughout this process can use those templates to register data, capture results, create inventory, and just record experiments as you would. From there, we'll take your feedback, and because we're iterative, we'll reconfigure and test again until we get to a point where we're happy and confident that this solution is going to work for your current processes and maybe helps expand on those processes so the future state is better than the past state. And for your customer go live, we're going to offer admin trainings and advanced trainings for your super users. We want to identify key folks within your organization who are going to be Benchling power users who will be able to answer the questions from your team as they come up, and we'll train them really well. Next, we'll do an end user training on the full platform and then template specific trainings for your individual teams. So if you have a team that's doing seed train production or a team that's doing plasmid design, they're probably doing pretty different things in Benchling. They'll both be trained on how to use the platform on the whole, but we can do more specific training for those templates so that your users are very capable and very confident in our platform. Following this, we'll have a go live for all users in production, and then we'll have ongoing support and of course, keep you appraised of the Benchling roadmap so that as new functionality comes out that could help you with your use case and some of your issues, we can offer that to you and continue to make the solution better as we go. Going a little bit deeper into this, if we look at the initial discovery, when we're sort of understanding that present, this is when we're going to be consolidating those many data sources, softwares, and steps into a streamlined process. So we're going to want to see your lab notebooks. How do you record data today? Do you use templates, and what do they look like? Hopefully your freezer is in better shape than this one, but we'll want to understand where things are, where they're located, and help you to if someone's looking for a cell line in the system, be able to find it. If someone needs a plasmid in the system, we can tell you how much you have as long as you're keeping that inventory solution up to date. Finally, we'll want to know those spreadsheets. We'll look at those column headers and we'll try to say, if this is a spreadsheet to track cell lines that they're using now, let's map that directly into Benchling, and then let's add on some other pieces of the Benchling functionality. Like now that we can track on a cell line the specific plasmid sequence and link to that, let's make sure that we do that in our system. In terms of building out an initial data model, this is where we don't have to start from scratch. And so as part of our best practices, we've modeled what a registry should look like. So each of the squares in this diagram is indicating something like a plasmid or a cell line, perhaps a virus for getting the plasmid into a cell line. And so we've had that kind of sussed out, and we can start you there. And we'll talk about how our best practices work and then figure out what's going to work for you. This will save us some meetings, and we can use those meetings to really get specific on the more intricate details of your process. Finally, once we built out that data model and understood your workflows, we're going to go about creating those templates to capture your processes. A template here for master cell bank preparation, like Wendy showed, might include some tasks that you can check off, some visual diagrams that you can drop in, and then there'll be things like registration tables where you can register new entities or cell banks into the system, or capture results perhaps with a result table, or perhaps after you've made something like a new cell bank, you can use a transfer table in our system to create the inventory of that and where that's going to live. So we'll work with your teams to build out these templates, first understanding your data, next using that baseline data model, then expanding on it, and finally putting that all together in these templates for you to use such that your team is able to enter data into the system and really get the most bang for your buck from the Benchling product. The Benchling professional service team, we love doing this. It's really fun to meet new clients and to take on new use cases. And we're really excited, when a new team joins. So we're looking forward to some of the folks on this call joining Benchling soon. Thank you.
Thanks, Sean. All right, everyone. We'll start with the Q&A. I'll kind of combine one from Sebastian and David regarding instruments. So can Benchling connect with instruments first of all, or is it only file-based? No, we definitely connect with instruments, but also a question from David is it through the API, and can we display data in the ELN page for a later review? I'll hand this one off to Prim. I think Prim also maybe a good thing to cover is on top of instrument integrations, what the most common integrations are that we see for cell line development.
Mm-hmm. Sure. So Benchling has the Benchling for Lab Automation product, plus the REST APIs on our developer platform. Those enable us to integrate with with a bunch of different analytical instruments and lab automation equipment. So some examples of integrations we have done are like Hamilton liquid handlers. We have done PA and TCAN liquid handlers. In terms of analytical instruments, we have a bunch of different integrations like plate readers and different analytical instruments. So I think the REST API, because of the open nature of integration, it allows us to integrate with a diverse set of different analytical instruments.
Okay, great. Wendy, this one's for you. Is when you were demoing, we got a question, is the software 21 CFR compliant?
Yes, actually, Benchline is designed for regulatory compliance with FDA 21 CFR Part 11. So we have audit trails, electronic signatures, electronic record support. And we actually adding to that question, something related to a validated cloud. We just launched the offering for a validated cloud in Benchline to support those teams working in a GXP environment. I don't know if Prem, do you want to add something about that?
Yeah, sure. Yeah, so with the validated cloud offering, we now offer a comprehensive package for you to actually perform the IQ, OQ, PQ validation to meet your specific needs. Yeah, so we have the full like update on how we like release a product and helping you support your validation requirements with the new validated cloud product. Actually, it's more like a service, not product, but it's more like a service. Mm-hmm.
Great, thanks both. We have a question coming in is, how can a core facility use this application? For example, flow cytometry. Sean, I think you'll be able to answer this one.
Yeah, I can take this one. So we see a lot of applications of Benchling for core facilities. And so what would normally happen is, there's going to be flow run against an entity. So whatever that biologic is would be registered in Benchling. And then the people that need the flow cytometry done would place a request to the flow cytometry team. That request could come in and could be triaged by the team. And the execution of that would typically happen in some sort of a notebook template. So the notebook template would be where you could type out what you did for that flow cytometry experiment. And then in a structured table, you would capture the results of that flow. That might be an image, it might be some numerical values, and all that could come in together. Once the request is complete, you would complete the entry. The user who initially requested that would be notified that the request was completed, and they can go and take a look at their data. That data would either be available on the request, or they could select the entity initially that they requested on. So maybe I needed flow against some cells. If I selected that, I would see the request panel there, or the result panel there of all of that result data. And I could then, with that data, do what I need with it.
Awesome, thanks. He sent a follow-up question is, they must have an account, which is, yes, you will have.
Yeah, so in the way that I just described it, yes, because you're capturing that flow data against another entity in Benchling. There are ways to do this if the requesters do not have accounts. But in that case, we're going to have to change the flow slightly such that someone is inputting what the result data is being tied to in this system.
Awesome, thanks, Sean. This one is for you, Wendy. Is what is Benchling permissions systems like? Oh, I'm sorry, Sean. I think I'll give this one to you. Such as, can you edit notebook templates, sequences, etc., or what are their permission systems for those?
Yeah, so the permission in Benchling is project-based. So if a sequence lives within a certain project, then whoever has access to that project and their level of access will determine what they're able to do. As an example, you may have a team that is really locked down, and so not that many folks at the company are able to see what's going on in that project. And if there was a sequence stored in that project and you went to look at it, you didn't have access, you might not see it. On the other hand of things, we like to make Benchling, if we're configuring, we want to make it as transparent as possible so we can kind of erase those data silos. But what this would mean is that you'll just have to set project level permissions. Our typical permissions are read access, write access, admin access, or none. We can get very granular with those permissions and create custom sets for your company, and which will allow folks to look at a sequence and register new sequences, but maybe not edit a sequence, or interact with the inventory, but maybe not interact with the registered item. And so we can get really granular with that during the course of the implementation.
Perfect, thanks. So I don't see any more questions coming in. We are getting close to time anyways. And just as an FYI, if you're a new user, you can request a demo right away at benchling.com/request-demo. We will be reaching out to those who said yes in the poll directly shortly after the webinar. For existing users and customers of Benchling, if you have any questions about today's presentation, please reach out to your customer success rep directly. And as always, please check out future webinars and events on benchling.com/events. And I hope everyone, yes.
Still, there's a question from Rachel. I think that's the
Oh, sure, I missed that one.
Yeah, so let me answer that question. So the question is like, how accessible is the data behind the scenes? Like, this particular user uses like a LIMS warehouse system for to do advanced analytical work. So I think with Benchling, just to reiterate, right, we have an open developer platform, and we offer like three ways of accessing your data. Like one is through the REST APIs that I mentioned. The second new type of integration we offer is called events. So events helps you like react to different actions on Benchling. And the third type, which I think you're referring here, is the data warehouse. So we do have a data warehouse where data is like synced on the backend, and then you can actually access that data warehouse directly and hook it up to advanced BI analytics tools like maybe Tableau or Spotfire and run your analysis that way as well. So yes, those are the three ways we allow you to access your data on the backend.
Okay, I'm pretty sure I don't see any more questions coming in, and thanks for stopping me there, Prim. Once again, I hope everyone has a wonderful day, and thank you so much for being here for this webinar. Take care. Bye-bye.